Hello, I welcome you to study session 10 of biochemistry, which will discuss protein and amino acid metabolism. Having learnt about protein and amino acid in the previous lectures, it is now time to learn about protein and amino acid metabolism. This study session will provide you with the knowledge of how the body digests proteins and how the urea cycle prevents toxic concentration of nitrogen, glucogenic, and ketogenic amino acids, and you'll be able to explain protein disorder. Moving to the introduction, in the mammalian body, proteins are not stored for later use. Excess proteins ingested in humans' body must be converted into glucose or triglycerides and used to supply energy or build energy reserves. Although the body can synthesize proteins from amino acids, food is an important source of those amino acids, especially because humans cannot synthesize all of the 20 amino acids used to build proteins. At the end of this study session, you'll be able to describe how the body digests proteins. You should be able to explain how the urea cycle prevents toxic concentration of nitrogen. You're able to discuss glucogenic and ketogenic amino acids and explain protein disorder. Digestion of protein. The digestion of protein begins in the mouth. When protein-rich foods enter the stomach, they mix with enzyme pepsin and hydrochloric acid. The hydrochloric acid produces an environmental pH of 1.5 to 3.5 that denatures proteins in the food. Pepsin cuts protein into smaller polypeptide and constituent amino acids. When the food gastric juice mixture, that is the chyme, enters the small intestine, the pancreas releases sodium bicarbonate to neutralize the acid, that is the HCl, which protects the lining of the intestine. The small intestine also releases digestive hormones called secretin and cholecystokinin, which stimulates digestive processes to break down the proteins further. Secretin also stimulates the pancreas to release sodium bicarbonate. The pancreas releases most of the digestive enzymes, including the proteases trypsin, chymotrypsin, and elastase, which aid protein digestion. Together, all of these enzymes break complex proteins into smaller individual amino acids. Once released into the small intestine, an enzyme found in the wall of the small intestine called enterokinase binds to trypsinogen and converts it into its active form, which is trypsin. Trypsin then binds to chymotrypsin to convert it to the active chymotrypsin. Trypsin and chymotrypsin break down large protein molecules into smaller peptides, which is a process called proteolysis. These smaller peptides are catabolized into their constituent amino acids, which are transported across the apical surface of the intestinal mucosa in a process that is mediated by sodium amino acid transporter. Moving to urea cycle. The urea cycle is a set of biochemical reactions that produces urea from the ammonium ions in order to prevent the toxic level of ammonium in the body. It occurs primarily in the liver and to a lesser extent in the kidney. Prior to the urea cycle, ammonium ions are produced from the breakdown of amino acids. In these reactions, an amine group or ammonium ion from the amino acid is exchanged with a keto group on another molecule. This transamination event creates a molecule that is necessary for the Krebs cycle and an ammonium ion that enters into the urea cycle to be eliminated. In the urea cycle, ammonium combined with CO2 resulting in urea and water. The urea is eliminated through the kidneys in the urine. Amino acids as a source of energy. Amino acids can also be used as a source of energy, especially in times of starvation, because the processing of amino acid results in the creation of metabolic intermediates, including pyruvate, acetyl-CoA, acetoacetate, and alpha-ketoglutarate. Amino acids can serve as a source of energy production through the Krebs cycle. Glucogenic and ketogenic amino acids. Amino acids can be classified as glucogenic, ketogenic, or both, based on which of the 7 intermediates are produced during their catabolism. Amino acid whose catabolism yields pyruvate or one of the intermediates of the citric acid cycle are termed glucogenic, while amino acids whose catabolism yields either acetoacetate or one of its precursors, are termed ketogenic. Protein disorders. Pyruvate dehydrogenase is the enzyme that converts pyruvate into acetyl-CoA, the molecule necessary to begin the Krebs cycle to produce ATP. With low levels of this pyruvate dehydrogenase complex, which is the PDC, pyruvate dehydrogenase complex, the rate of cycling through the Krebs cycle is dramatically reduced. This results in a decrease in the total amount of energy that is produced by the cells of the body. The pyruvate dehydrogenase complex deficiency results in a neurodegenerative disease that ranges in severity depending on the level of the pyruvate dehydrogenase complex enzyme. It may cause developmental defects, muscle spasms, and death. And treatment can include diet modification, vitamin supplementation, and gene therapy. However, damage to the central nervous system usually cannot be reversed. People afflicted with PKU, that is phenylketonuria, lack sufficient activity of the enzyme phenylalanine hydroxylase and are therefore unable to break down phenylalanine into tyrosine adequately. Because of this, levels of phenylalanine rise to toxic levels in the body, which results in damage to the central nervous system and brain. Symptoms include delayed neurological development, hyperactivity, mental retardation, seizures, skin rash, tremors, uncontrolled movement of the arms and legs. Thanks for listening. Bye.